Good morning. Today we're going to kind of go into genetic engineering. So kind of what can we do with our DNA now that we know what genes we have and things like that. So what genetic engineering is it's manipulating genes for practical purposes. And usually there are two reasons why DNA is genetically engineered. The first is for medicinal reasons, trying to create some sort of product that we need in the medical community. One of the more well-known is making human insulin for diabetics. We've actually been able to have bacteria produce this by inserting human genes. The second reason is agriculture. This has come under fire a lot recently with the genetically modified organisms that are being produced. Although most of the genetically modified plants aren't generally used for human consumption. At the very end, when you have taken DNA and genetically engineered it, you end up with what's called recombinant DNA, or DNA that has two different parts, if you will. Comes from two different sources. For the medicinal purposes, we will use human DNA and insert it into a bacteria so that the bacteria can manufacture whatever it is that we want it to produce, like we said earlier, so that it can produce more insulin so that the diabetics can actually get human insulin. We're just using the bacteria as essentially a factory for it. So the first people to develop recombinant DNA was Stanley Cohen and Herbert Boyer. They took genes that coded for ribosomal RNA from a frog and then inserted that into an E. coli genome. So you ended up with bacteria that could produce frog ribosomal RNA. Sorry, my allergies are kind of going crazy today. All right, so the first thing that will happen, there's 4 steps, basic steps when DNA is genetically engineered. The first thing that you're going to have to do is cut your DNA. The picture down here is bacterial DNA. You can tell it's bacterial DNA because it's in the shape of a circle, in that plasmid shape. It's what a circle of DNA is, and we call it plasmid DNA. And so it's still double helix, but it's just in a circle instead of strands. So basically what they're going to do is they're going to cut out the gene of interest and then the vector's DNA. So whoever's going to get that gene, whoever's going to have to produce that gene. Generally, it's going to be bacteria. The way they're, I'm going to get ahead of myself, I'm sorry. The plasmid DNA can replicate independently. So they tend to be the preferred choice. The way they cut out that gene and cut open the vector's DNA is they use these things called restriction enzymes. So they are going to be, the restriction enzymes are programmed to look for a certain base sequence, and then they cut off the DNA at that particular point. They leave what's called a sticky end because when they do the cutting, they will cut maybe 2 bases deep into the right side of the strand, but then 4 or 5 bases deep into the left side, and so you end up with uneven DNA strands. So basically, on one side, you've got a couple of bases just randomly sticking out. But that allows for it to pick up the gene of interest. So they will put these restriction enzymes in with a vector DNA to get it to, well, at the vector's DNA, and then they'll also do this with the DNA that has the gene that you want. Then what they're going to do is they're going to put those two together, and since they both have sticky ends, they'll naturally, think of it like Velcro, they will naturally stick together. On Monday, I'll have a picture for you. So the second step is the forming of that recombinant DNA with those sticky ends. So like I said, they're going to join the vector's DNA with the gene of interest. They're going to combine them. They'll mix it in with DNA ligase to help bond that DNA, since its job is, it's basically like connecting those Okazaki fragments, except we're joining the the vector's DNA with the gene's DNA. The third step is going to be cloning. So with cloning, you're going to end up with, you're going to just replicate the DNA over and over and over again. So you have many cells with that new recombinant DNA. The fourth step is the screening step, which tends to be the more difficult step. So genetic engineers will screen those cells to see which ones took up the recombinant DNA. Usually, they're going to add an antibiotic to the cultures to see if those cells have taken up the DNA, because usually the recombinant DNA is resistant to the tetracycline. So it's going to kill off any cell that doesn't have the recombinant DNA. So then what they'll do is they've killed off the cells that don't have the recombinant DNA. So, because not all of the ones that were cloned end up with that recombinant DNA. And so what happens is then they will then isolate those by taking a little inoculating loop, taking up those cells that have the resistance to the tetracycline, the ones that have the recombinant DNA, and then they are allowed to replicate and form a colony. So then you would have a culture of bacteria that could produce insulin, if that's what your recombinant DNA was. Like I said, Monday I'll have a picture for you for the sticky ends to kind of give you a better idea as to what you're looking at. So, all right, well, that pretty much concludes the end of this part of it. We'll talk about how they do some more screening on Monday. So, hope you have a great weekend.